And I will start with the topic of conducting experiments. Some things that we haven't discussed so far, but first I want to remind you about the two basic ways of assigning participants to experimental conditions. For the last two classes, we have discussed that we can assign participants using a between groups design or independent groups design, or we can assign participants using a repeated measures design. Do you remember what is the difference between those two designs? I hope that you do remember. If we have an independent groups design, each participant takes part in only one experimental condition. And then we compare the results between different groups of participants. And because of that, it is called a between groups design. And we discussed that it is really important to use a random assignment to experimental conditions. If we are using independent groups design. And last time we discussed another technique that could be used to assign participants, not using random assignment, but using match groups. When we are using match groups, first we match the participants on some variable that is related to the outcome of the experiment. We form some pairs or groups of participants that are matched on that variable, and then we randomly assign members of this group to the experimental conditions. Again, we have random assignment. And we discussed the repeated measures design, in which each participant takes part in each experimental condition. And it is also called a within subjects design because we make comparison within the same group of participants. And last time we discussed different counterbalancing techniques. Those are techniques that are used to control for possible confoundings, because we discussed that this type of design has a lot of advantages, but it also has the disadvantage that because each participant takes part in all of the experimental conditions, you can have the so-called order effects. So the performance improves or gets worse because of the time spent in the experiment. It could have two different forms. So we can have the so-called learning effect, so people get better with time, or we can have the so-called fatigue effect. So people get worse and worse in the task because of the time they spent in the task. Or we can have the so-called carryover effects. So when the manipulation in one of the experimental conditions is transferred to the other experimental condition. And last time we discussed different counterbalancing techniques that we can use in order to control for possible confoundings. And they're called counterbalancing because we are not able to completely get rid of the error, but we can try to distribute that error among all experimental conditions. Do you remember some of the counterbalancing techniques that we have discussed last time? We have discussed a lot of them. Somebody is writing in the chat.
Time randomization or maybe stratification. But speaking of my previous studies.
Okay, what do you mean by stratification?
The sample selection and how we select it.
Okay, but this is a technique that is used for choosing the sample that you are going to use. But I'm asking about the techniques that could be used if you have chosen the sample already. You have designed repeated measures design, so each participant is going to take part in all of the possible experimental conditions. And then there are some techniques that could be used in order to counterbalance the error. And we discussed them last time. So we could have counterbalancing techniques within individuals or between individuals. Does it make you remember some of those techniques?
For example, BBA,
Yes, APDA.
block randomization.
Mhm.
Mm,
So these two are counterbalancing techniques within individuals. So within each individual, we are presenting each experimental condition several times. Like you said, ABBA or block randomization. But if it is not possible to present each experimental condition several times to each participant, then we should use other techniques. Do you remember which are they? So the first one was to present the experimental conditions in all possible orders of presentation. Do you remember that one?
yes, and the other one was selected order or Latin square, I think.
Yeah. So if it's not possible to use all of the possible presentation orders, we can select some of them. We can select them at random, or we can use Latin square to select some of the experimental conditions. And I'm not sure how many of you remember those techniques. Do you have questions about them, or you didn't have time to look at them? Is there a need to explain something again? I think you have a lot of explanations in the book chapters that you have uploaded in Moodle, and also you can listen to the previous presentation. But this is really important. So in a repeated measures design, if you don't use at least one counterbalancing technique, then your experiment is confounded, and then you cannot make any conclusions from such an experiment. So it is really, really important to use counterbalancing techniques in repeated measures design. And it's because of that, it is important for you to know the available techniques and to be able to choose one of them. Maybe later when we do the exercises, we can speak about them again. But what I want to talk about now is the so-called control condition. This is something that is already a common knowledge. Maybe people use the term also in everyday language, but in experimental designs, control condition means something really specific. And this control condition is the experimental condition in which there is no influence of the independent variable. So the independent variable in that condition is 0. Then it is called a control condition. And why do we use control conditions? Because we want to have a baseline to judge and to compare our results to that baseline. One example of a control condition could be given in an experiment in which we are interested in the effect of noise on studying. And then if our experimental group studies while a loud noise is present, then the control group should be studying the same material for the same duration, but in a quiet setting without any noise. Because noise is missing in the control condition, because of that, it is called a control condition. But let's get that example. Again, you're studying memory, and participants are required to learn lists of words. And the experimenter is interested in how learning one list of words interferes with learning another list of words. And then the experimenter has an experimental group. Participants in that group first learn list A, then they learn list B, and they're tested on the first list of words. They're tested on list A. And the experimenter wants to check if learning the second list of words, list B, interferes with the studying of the first one, the studying of list A. So for the experimental group, we have list A, list B, and then the participants are tested on list A. What could be a control condition in such an experiment? So here the independent variable is the presence of the second list, just to make it clear. How would you suggest to have a control condition in that experiment?
The presence of only one list, so not A, B, and just one list.
So if we have list A, and then the participants are tested on that list. But,
Yeah.
there could be a possible confounding here, because it is going to, so when participants learn list A and they're immediately tested on that list, and in the experimental group, they study list A, then they spend some time studying list B, and then they're tested on list A. And even if we get that they are remembering less number of words, then one alternative explanation could be that maybe just more time elapsed between studying the list and testing on that list. So what can be done then?
Well, then put the time between list learning and testing to be the same no matter what. So if you're going to learn list A and then be tested on A, make it 24 hours. So if you learned it at 9 a.m., you get tested at it on 9 a.m. the next day. And the second group will learn list A in the morning, and then list B in the afternoon, and then be tested on list A at 09 the next morning still.
Okay, so this is one possible way to deal with that problem. Another possibility, if you don't want the participants to spend 2 days in the experiment, I thought that you were proposing that is just to provide some time between, for the control group, I mean, between learning the list A and testing on the list A. Let's say that studying list B takes 15 minutes, then you can provide a 15 minutes break between studying the list A and testing on the list A. Do you like that suggestion?
It could work.
It could work, but again, you can have more concerns. So here there is another suggestion how it could be done. So for the control group, they can first learn list A, then do some arithmetic tasks, and then they are going to be tested on list A. Why they have included arithmetic task in the control group?
To simulate the fact that their mind is engaged between the learning and the listing.
Yes, and it is important to have their mind engaged, because maybe if you just provide some free time, they're going to repeat the words that they have studied already. And this is going to improve their test results. So there is a comment in the chat. Okay, yes, they can do something else, but reading would not be a good idea, because in reading, again, you are exposed to words, because usually it is another task that is chosen that is not similar to the task that is used in the experimental group. I'm just giving this as an example that sometimes it is not so easy to be able to find out which is the control group or the control condition that is suitable for your experiment, because it is really easy to say, okay, in the control condition there will be no effect of the independent variable. But then when you start thinking about the details of the study, it seems that it is not so easy to do it. And I'm not sure who raised the question of sample selection. I'm not sure if he's present now, but this is also something important that that you should consider. In general, in statistics, it is said that you should do probability samplings. So a really random sample should be randomly drawn from the population. So you should be able to randomly draw participants from the whole population in Bulgaria. Usually it is not possible. And when they're doing some survey research, not in the field in psychology, but in the field of sociology, for example, sometimes stratified samples are used, as it was mentioned. So in a stratified sample, you try to represent all possible, you try to match the demographic structure of the population based on gender, age, education, places of living, and so on. However, in psychological research, what is most often used is the so-called convenience sampling. Studies are done on samples that are readily available, usually students, usually undergraduate students. I think I have told you that some people joke that psychology is a science about students in psychology, because most of the experiments in psychology are done with samples consisting of students, and especially psychology students. And in fact, that convenience sampling is not a problem for the internal validity of the experiment. So it is not a problem of being able to find the effect of the independent variable on the dependent variable. It is a problem concerning the external validity of the findings. So the question that we should ask is, can you apply the findings that we got, not only for students in psychology, but also for all other people? So this is the external validity of the experiments, and using convenience sampling is a problem, and it's a threat to the external validity. There is also, you can have it like a joke, but it is not a joke, that even if you try to include participants that are not students in psychology, often what we get is the so-called weird sample. Weird stands from white, educated, industrialized countries, rich people, and with democratic orientation, democratic political orientation. And this is again a threat to the external validity of the findings. And now a lot of efforts are made to include more diverse samples in research, using online experiments, using replications in different countries, in different cultures. But still, this is a problem in psychological research, that samples are very often convenient samples. And then the next topic that I want to discuss is how we manipulate the independent variable. We always have some operational definitions of the independent variable. We should find a way to provide different experimental conditions for the participants. And there are two types of manipulation of the independent variable. One of them is called straightforward manipulations. In such type of procedures, what we do is that we manipulate the independent variable straightforward with using different instructions, with using different stimuli. We change something in the stimuli, let's say the color, the noise, or we provide specific instructions in written form, or like verbal instructions. But it is clear what we manipulate in our experiment, and we are pretty sure that we are making the difference that we want to make between the experimental conditions. If you have thought about the example with global or local features that we had last time, and that is available online, then this is a straightforward manipulation. So you're presenting a stimulus that is either a global letter H or another letter that is composed of small letters H. This example of a straightforward manipulation. But sometimes another type of manipulation is used. This is the so-called stage manipulation, when the experimenter very often uses the so-called confederates. Those are helpers to the experimenter that design some social situation in many cases, in which they try to create some psychological state like anger or frustration. And usually such manipulations are used in real life settings, like you can do it on the street or while participants are waiting to enter the lab. And then it is not sure if your manipulation is successful. It also applies to the straightforward manipulations, but especially for the stage manipulations, it is really important to include the so-called manipulation check in your experiments. And this step in conducting the experiments is sometimes skipped by the students. So you should design a procedure that is aimed to measure if you are in fact able to successfully manipulate the independent variable. Because you can use, let's say, videos trying to induce different emotional states in the participants, but you should have a way to check if this is actually going on. Maybe you are not sure if the video actually made the participants feel happy or sad or angry. So a manipulation check is a procedure that is used during the experiment, and by using that procedure you want to check whether the independent variable manipulation was in fact successful, if you have chosen the correct operational definition of your independent variable. And here is another example. If you try to manipulate the anxiety, you want to have one group of participants that is feeling anxiety and the other one that is not. And then you should check if participants in the high anxiety group really felt more anxious than those in the low anxiety condition. And how can you check that? I'm looking at the time, and I'm trying to save some time because of that I'm going to tell you how you can do it. You can include self-report measures of anxiety. You can have a rating scale from 1 to 5, asking participants to rate themselves how anxious did they feel during the experiment. Or you can have a behavioral measure of anxiety. You can count how many times they have moved their arm or hand. Or you can have a physiological measure of anxiety. You can have their skin conductance measured or their blood pressure measured. And then you can have a manipulation check that your manipulation was successful. And this is something really important in conducting the experiments, because if you are manipulating some physical appearance of the stimuli on the screen, you should you are confident that you are doing the manipulation. But if you try to induce especially some emotional states, you should be somehow confident that your manipulation was successful. And manipulation checks are sometimes done during the so-called pilot studies. So pilot studies are run with small samples before you start the actual experiments. And then you can check if your manipulations are working in the way that you expect them to work. And if they're not, then you can check, you can change the procedures that you use to manipulate the independent variable. And it is also important to include manipulation checks during your actual experiments. But as it might reveal the goal of the experiment, or it may distract the participants, usually it is done during the debriefing phase of the study. Debriefing is the part of the study when you thank the participants. Usually you explain the real goal of the study, but you can also use that time to ask the participants about the independent variable that you try to manipulate, or different questions if they have guessed the goal of the study, and so on. We are going to speak about this later. And then this topic about how we measure the dependent variable, that should also be decided when you are planning a study. You remember that you should decide between 3 different types of measures. You should decide if you are going to use a self-report measure, a behavioral measure, or a physiological measure. You should choose measures that are sensitive. So you should be able to detect small differences in the performance of the participants. And you should try to avoid ceiling or floor effects. Ceiling effect means that everybody performs getting the maximum score, and floor effect is when the task is always so difficult that everybody is scoring really low. And we discussed such concepts as reliability, validity of the measures. And I have mentioned several times that in the experiments, the simple examples that we are looking at, usually there is just one dependent variable, but in real experiments, usually there is more than one dependent variable. You can measure several dependent variables at one and the same time, or you can measure one and the same dependent variable using different types of measures. And again, all other variables, we manipulate the independent variable, we measure the dependent variable, we check if our manipulation is successful, and all other variables that are external to the experiment should be controlled. And all those variables are physical variables, so variables related to the physical properties of the stimuli, of the environment, and so on. Individual differences, social differences, everything should be controlled for. And the techniques that are used to control for such variables are elimination. We can try to eliminate an external variable, or we can try to keep it at a constant level. And if it is not possible, we should use randomization or counterbalancing. And because of that, it is really important to know all of the counterbalancing techniques that could be used. And when conducting experiments, there are also additional considerations. We should take into account the participants' expectations and also the experimenter expectations. Participants' expectations about the study usually are defined in two groups that have an effect on the results of the experiment. First, you could have the so-called demand characteristics. This is a problem that could arise in a study if participants have some expectations about the goals or about the hypothesis of the study. And then they could change their behavior, they could change their performance in order to confirm your hypothesis if they want to help you, or they can change their behavior in order to refute the hypothesis if they guess what is your hypothesis. And also another problem is the so-called placebo effect. I guess that most of you are familiar with that placebo effect because it is discussed a lot when it comes to drugs and testing the effect of drugs. But also in psychological experiments, people can change their behavior, can change their feelings, they can change what they actually experience, not because of the manipulation of the independent variable that you are doing, but because they expect that an effect is going to happen. And it is important to notice that participants could change, in fact, their perceptions. They can start perceiving something as better or more painful and so on based on their expectations. And to deal with such participants' expectations, very often the goal or the hypothesis is not taught in the beginning to the participants. Usually some type of deception is used. Participants are told that something else is being studied in order not to change their behavior. And also when you are thinking about the dependent variable, you can try to measure the dependent variable in an unobtrusive measure so the participants are not aware that they're being measured, or you can try to put the dependent variable, let's say a question that you are asking among a set of other so-called fewer items, among some other questions that are not relevant for your study, you are not interested in the results about these questions, but you are using them to be able to disguise what you are actually measuring. And also what is done is that you can use the so-called placebo groups. So you can use a group in which the participants think that they're receiving the intended manipulation. If it is a drug study, all of the participants think that they receive the actual drug, but in fact, half of them are not receiving a drug, but they're receiving some sugar pills, for example. But it also applies to many studies in psychology. If you are studying, let's say, taste perception, you probably know that there are a lot of studies about Pepsi and Coke. If people are asked what is their favorite drink and they say Coke, if you present them two types of drinks that are labeled Coke and Pepsi, then participants are going to prefer the drink that they actually prefer. But if you present the drinks in cups that are not labeled, and you ask them to taste each of the drinks and to state which one they prefer, usually half of the participants change their preference, and they do not actually state that they prefer it. And using placebo groups is really important in many experiments if you expect that participants can change their performance based on the fact that they know what condition they're in. There is another really funny study using beer and vinegar, in which they put some drops of vinegar in the beer, and they asked the participants to taste the beer and to rate their liking. And it is shown that when participants know that there is vinegar in the beer, they don't like it. But if they don't know that there is vinegar in the beer, they like it more. So now we don't have time, but maybe next time or in some of the other classes, I can show you more about that study. It is a funny one, because it also explores if just the answers are changed, or also if the real perceptions of the participants are changed. Because there are a lot of studies in which it is shown that the actual perceptions, the actual sensation is changed. So they're not just biasing their answers like when we are speaking about demand characteristics, they're just trying to please you, and because of that, they change their responses, but they actually perceive differently what they're drinking. And such experiments are called blind experiments if the participant is not aware which is the experimental condition that is going on. It is called a blind experiment. Like I said, using, for example, unlabeled cups or unlabeled pills and so on. But also we should pay attention to the experimenter's expectations. So if the experimenter has an expectation how the participants should respond, they could bias the result. So they can intentionally, or most often unintentionally, treat the participants differently in different experimental conditions of the study. If they know that in one of the conditions there is alcohol involved, maybe they're going to read the instructions slowly or slower to the participants. Or if you are doing video recordings and then somebody has to code for the results, let's say how many times the participant moved his hand or arm, then if the experimenter knows which is the experimental condition, this could bias the results and the rating. During the first lectures, we discussed the clever Hans effect. I hope that you all remember that effect, that the experimenter's knowledge about the correct response somehow can cue the participant in providing that correct response. And there is also another really famous study. It is the study by Rosenthal conducted in 1966, in which graduate students were asked to train rats that were described as coming from either maize bright or maize dough genetic strains. So they were told that some of the rats are bright and they are able to learn really fast, while the other rats are dull and they are not able to learn fast. And what is found in that study is that, in fact, bright rats did perform better than the dull rats. However, in that study, the actual subjects were the experimenters. In fact, the rats were the same, so there was no difference in the rats. They were not coming from different genetic strains. This is the title of the paper that was published. If you're interested, I can upload it. But in fact, the participants in that study were the experimenters, and half of them were told that they're going to train bright rats, half of them are told that they're going to train dull rats. So I would try to show you here. So those are the results for the, okay, it's not successful. That drawing never managed it, but I guess that you can see here what are the results measured in a number of correct responses when the experimenters are told that the rats are bright, and on the right, this is something going on with that. I'm sorry, I have to reopen the presentation. Okay, here the column that is labeled bright is the results for the rats trained by experimenters that are told that the rats are bright. And the next column that is called dull are the results of the rats that are trained by experimenters that are told that the rats are dull. And here at the bottom, you can see the average number of correct responses for one group of experimenters that were told that the rats are bright, and for the other group. So it is a classical study, a classic example how providing different information to the experimenters can bias the outcome of the study. And because of that, usually the experimenters, the people actually collecting data, are not the same people that have designed the study. And the actual hypothesis of the experiment, in fact, is not taught to the experimenters, because we don't want to bias their results. Also, it is really important to have experimenters that are trained really well, if possible, to use instructions that are written and that are read aloud by the experimenter without changes. Experimenters are not allowed to explain the instructions. They should read, and if needed, to repeat the instructions. We could try to use computers and computerized presentation of the stimuli and computerized scoring of the results, again to prevent that experimenter's bias. As I told you, experimenters should be unaware of the hypotheses that are being investigated. And also, the experimenters should be blind, so they should have no information in which condition the participant is taking part in. And then we can have the so-called double-blind experiments, in which neither the participant nor the experimenter knows which is the experimental condition that is being administered at the moment. It is done, as I said, using computers, in which the computer randomly presents the experimental condition, or by using two experimenters. One is, let's say, pouring the drinks in unlabeled cups and just putting some codes on the cups, and the other experimenter is administering the study. And those participants' expectations and experimenter expectations should be really controlled for, because they could bias a lot your results. And the final slides are about the pilot studies. I already mentioned them, that it is a good practice to have some practice runs with a small number of participants to check if all of the instructions are understandable, if the manipulation is successful, and so on. And also, at the end of the study, you should have a so-called debriefing session. We are going to discuss it in relation to the ethical aspects of conducting the study, but also this is the time in which you can check for participants' expectations. You can ask them what they thought was the goal of the study, what was the hypothesis, and in such a way you can check if some of the participants in fact have guessed your hypothesis, and maybe there was demand characteristics present. And there is a comment in the chat. Maybe Nina, if you want, you can turn on your microphone. So when you bought a beer from my country, poured it in a bottle of a German beer, gave it to my friends and told that it's from Germany, all of them loved it and said that there is a big difference in quality. Yeah, so it happens a lot, and there are a lot of studies with beer and also with wine. If you change the bottle of the wine to be more expensive and to look more expensive, people like it more, and it's not an interruption, it is just the topic that we are discussing at the moment. So I think it is important to discuss such topics. And here is an example of a study that we can try to have an exercise on. The title of the study is Keep Your Fingers Crossed, How Superstition Improves Performance. Do you find that topic funny? I thought it was funny. It was cited in one of the textbooks, and I looked for that paper. And it seems funny, and the experiments are described in short. So I want to use that example to make an exercise. So superstitions, many people have superstitions that if they do something, they are going to be lucky. I don't know if you have such ideas. Maybe some of you have so-called lucky charms and so on that are going to help you in performance, let's say during the intermediate assignment. And the idea of that paper was that if we activate such superstitions, people are going to perform better. In fact, they had several experiments, but in the first experiment, you can read what is the idea. Experiment 1 was designed to examine for the first time whether activating a superstition improves subsequent performance. Specifically, we activated the superstitious concept of good luck by associating it with the bow participants used in a putting task. So here on the right, you can read how the study was conducted. I will give you several minutes to think about it. And what you should think about is, again, using the structure that you already know from the previous exercises. What is the hypothesis? What is the independent variable, the operational definition, the dependent variable? What is the type of design that is used in that study, independent group design or a repeated group design? And then we are going to discuss it. So I will give you several minutes to read it. Are you able to read it on the screen like that? I hope
Yes.
so.
We are able to.
Yeah, so just I will give you several minutes to read it. Do you find the topic funny? Yes, Stella said that this could be really practical. I agree, especially if the results show that it can improve performance.